Hello everyone. This is Dr. S. Abinaya from the Department of Biotechnology, Kaveri College for Women. Today my topic is on Introduction to Cell Culture. Let's begin with what is cell culture? Cell culture is nothing but the removal of cells from an animal or plant and their growth in an artificial controlled environment. It is nothing but rearing cells outside the body of any organism. So it is called as in vitro method. So the cells can be directly removed from the tissues and they can be cultured in a controlled environment. So this is about the history, how this cell culture has begun. It just began with the discovery of the term cell and how different scientists proposed the cell theory, and Rocks was the first one to introduce the method of cell culture. So number of scientists were involved in various steps in establishing cell cultures in artificial and some natural media. And this is a major breakthrough in the biotechnology research. So just this picture shows the types of cells in the body. So our organs contain different types of cells which are specific to the particular organ, like the bone cells, blood cells, muscle cells, skin cells, and nerve cells. Each are unique in their own way. And the stem cells are the cells where the other cells get differentiated to. So stem cells are the origin from which the other cells get differentiated with particular specificity. So in vitro cell culture is the technique which involves the rearing of cells outside the organism in a glass or some culture vessel or a petri dish in a controlled environment. So controlled environment refers to the environment which is completely controlled artificially with all the necessary requirements for the cell to thrive. So primary cell culture basically refers to the culture from where the tissue or the cell is directly collected from the animal and cultured first. So the preliminary step of cell culture is called the primary cell culture, whereas the secondary cell culture involves the subculturing of the primary cell culture where the cells established already. So cell line is nothing but after the first subculture, the primary cell culture becomes the cell line or a subclone. So it is the beginning of the lineage. It is the beginning of the cell line lineage. So there are two different types of cell lines, like finite cell lines and continuous cell lines. So normally the cells used to divide only for a limited number of times before losing their ability to proliferate. So after a particular point, they will reach senescence where they will not be dividing further. And this type of cells are called as finite cell lines, whereas the continuous cell lines have indefinite divisions. And a normal cell line can be converted into a continuous cell line by a process called. So let's see what is required for a cell culture basically. The basic cell culture facility requires a unique and very dedicated infrastructure which should be completely aseptic. It should be isolated from the other parts of the buildings, and it should have limited access to people, and the building can be constructed or the facility can be constructed in an area where there is minimal footfall. So this is the basic prerequisite of establishing a cell culture facility. So this picture shows the layout of aseptic environment which is required for a basic cell culture facility. So there will be designated areas which are separated from each other, like there is a separate preparation area and a corridor, a washing area, quarantine room, microscope room, hot room, cold room, storage room, and the working area. The working area should be completely maintained in a aseptic condition. So this is the basic layout of a cell culture facility. And the cell culture requires different consumables like culture vessels. And this picture shows the tea flasks, which is the most preliminary type of cell culture vessel. So it is available in different volumes based on the need. And the cells can also be cultured in different multiple plates. And the essential media is required. So media plays a very crucial role in the growth and morphology of the cells. So the medium can be supplemented with fetal bovine serum or some other serum, which provides nourishment like growth factors and other necessary factors for the proper thrival of cells. And there are so many consumables like serological pipettes in various volumes, and all the products should be disposed after a single use. Most of the products cannot be used for a multi times. And this is the pipette dispenser, but this will be used for aspiration and dispensing of the fluids. And this is the biosafety cabinet, which is the very most prerequisite of any culture facility. And also we have CO2 incubator. This is somewhat different from the normal incubator where the CO2 influx will be produced completely in order to maintain the gaseous exchange properly. And also we have an inverted microscope here. The special purpose of this inverted microscope is to view the adherent cells from the bottom of the flasks. The adherent cells tend to attach to the substratum or the bottom of the culture vessel. So the objective lens will be present at the bottom in case of inverted microscope, which facilitates the proper view of the cells. And these are the applications of cell culture, why we need cell culture. So this can be used for performing various studies on intracellular activities like cell cycle, differentiation, and metabolism. And it can also be used for elucidating the intracellular flux, hormonal receptor, signal transduction, and all. So most metabolic activities and the cellular progress can be studied using this cell culture facility. And also it can be used for production of number of pharmaceutical and therapeutic products like plasminogen, interferons, blood clotting factors, hormones, monoclonal antibodies, vaccines, etc. So it is the most preliminary and most advanced type of technology in biotechnology. Thank you.